Today's presentation is Fostering Pediatric Oncology Drug Development. Next slide, please. Our speakers are Lynn Georgopoulos, Amy Chung, and Larissa Wenning. Lynn is currently a Vice President of Regulatory Strategy at Certara. She has over 30 years of experience developing and providing strategic guidance and clinical and regulatory advice at all stages of product development. Prior to coming to Certara, Lynn was a Vice President of Regulatory Affairs and Pediatric Strategic Development at Synteract and Senior Vice President of Regulatory Affairs and Clinical Development at Kinderpharm. Amy is currently a Senior Director in the Integrated Drug Development Group at Certara. She has over a decade of experience in clinical development in the pharmaceutical industry. Dr. Chung earned her doctorate and completed a postdoctoral fellowship at the University of Manchester on structural identifiability and mechanistic modeling. Larissa is currently a distinguished scientist in quantitative pharmacology and pharmacometrics at Merck. She's also currently Vice Chair of the ASCPT Global Health Community. Dr. Wenning holds a doctorate in chemical engineering from the University of Wisconsin-Madison, and she conducted postdoctoral research at Northwestern University. Lynn, Amy, and Larissa, welcome to today's event. I'll now turn it over to Lynn to begin the presentation.
Thank you all for attending this webinar. We hope that you will find this information informative. Our goal for today is to provide you with a brief overview of the current landscape of pediatric cancers with respect to incidence, distribution, therapeutic approaches, and how that information will impact on how sponsors will approach compliance with the requirements of the Research to Accelerate Cures and Equity for Children Act, in short, the Race for Children Act. I will provide a brief summary of the statutory requirements and available resources. My colleague, Amy, will review the quantitative considerations to approaching pediatric drug development, and Larissa will then provide an exemplar case study on how quantitative methods were used to support pediatric labeling of Keytruda. So I thought it would be important to start this discussion with some statistics on the incidence of pediatric cancer in order to have an appreciation of the problem. We will start with a polling question. So in 2020, it is estimated that approximately 1.8 million new cases of cancer will be diagnosed in the United States. Please select the number of cases that you think occur in pediatrics. We'll give you a minute to reply. Okay, great. And the answer is less than 1%. So will be diagnosed with cancer in 2020. About 11,500 children between birth and 14 years of age will be diagnosed with cancer, and about another 5,800 adolescents between the ages of 15 and 19 years will be diagnosed with cancer. Although cancer in children is rare, it is the leading cause of death by disease past infancy among children in the United States. Childhood cancer incidence rates have slowly increased each year since at least 1975, which is really the point at which data was beginning to be collected. Between 2007 and 2016, rates have increased on an average of 0.8% annually, with the strongest increase in infants. And this data is actually consistent with global trends that have been reported. Having an appreciation for the incidence of pediatric cancers will be an important consideration when we start to talk about and thinking of our approach to designing clinical trials in this population. Since we have limited time today, I'm going to be focusing primarily my discussion on U.S. regulatory considerations, but certainly these programs need to be addressed on a global scale. Great, thank you. The data presented here is our incidence rates by the International Classification of Childhood Cancers. And this classification is really based on the tumor morphology and primary site, with an emphasis on the morphology rather than the emphasis on the primary sites, as we see in adult cancers. The three most frequent pediatric cancer groups really comprise leukemias, brain and central nervous system tumors, and lymphomas. The challenge is addressing pediatric cancers is that really pediatric cancer is really not one disease, but really comprises over 100 different types of cancers that are seen in these patients. I know this slide's a little bit busy, but I thought this would be helpful to really get an appreciation of the complexities of studying pediatric cancer, as there are, again, numerous subtypes. But adding to that complexity is really the distribution of cancers also varies significantly by age. So not only are there differences when we think about differences between adults and children, but also within the subgroups of the various pediatric age bands. So for instance, if we look at children diagnosed with cancer, the incidence of leukemias in children between the ages of 1 and 4 is more than twice that of adolescents, 15 to 19 years of age. And lymphomas rarely, for instance, occur in children under the age of 4, but almost really comprise about a quarter of all the cancers diagnosed in adolescents. If we drill down even further to a subgroup of malignant epithelial neoplasias and melanomas, we see the greatest incidence of cancers really arising from adolescents, really having derived from thyroid cancer. So really when we can begin to think about therapeutic approaches, study design, understanding the variability and incidence across the pediatric age ranges will be very important. Again, another busy slide and lots of data, but here, really, when we think about cancer therapies and assessment of response, we're really looking at the overall 5-year survival. And so we see here that obviously with the treatments that have been available thus far, we've been able to make a significant impact on the overall 5-year survival rates. And we have seen a dramatic decline, especially with the beginning of first therapies from the early 70s. And although the overall survival rate is about 84% overall, this rate varies considerably by age and by cancer type. And so there's still much work to be done, as noted in the reds that I've highlighted, where we see less than 75% response rate. So when we think about our treatment strategies, our current multimodal approach for treating pediatric cancers involves surgery, chemotherapy, radiation therapy, and stem cell transplantation. And as progress has been made in the understanding of the genomic landscapes of pediatric cancers, where malignancies that occur in children and adolescents can harbor some of the same molecular abnormalities as adults, we've seen an emergence of the use of targeted therapies. Unfortunately, some of the unwanted side effects were able to improve overall survival, but there are some significant side effects to some of the cancer treatments that actually may cause more harm to children than what we see in adults, because children's bodies are still growing, they're developing, and so a lot of these cancer treatments are more likely to affect developing organs. And so research has shown that more than 95% of childhood cancer survivors will actually have significant health-related issues by the time They are 45 years of age, primarily as a result of their initial cancer treatments, with about a third suffering severe chronic side effects, a third with some moderate health problems. So, you know, there really is a significant need for us to develop more effective therapies with less toxic effects. And this is really just the driver behind the RACE Act and moving to try to expedite alternative cancer therapies for children. So with that, I'm going to just move on to just a brief overview of the evolving US regulatory landscape to foster pediatric drug development. So we can go back to 1984, where the pediatric rule was enacted to really allow for the extrapolation of efficacy data from adults to children, obviously still evaluating safety and pharmacokinetics, but you know, this is not a new concept. In 1997, there was attempts to incentivize pediatric drug development by offering 6-month extensions of marketing exclusivity, but really this didn't do a whole lot to spur on pediatric drug development. In 1998, the pediatric rule was introduced and required pediatric assessments for drugs likely to be used by children, but this was subsequently declared invalid. So really the big change really came in around 2002, 2003 with the introduction of the Best Pharmaceuticals for Children Act, and that really provided some incentives for sponsors to conduct pediatric studies of drugs that the FDA believed may produce benefit in the pediatric population. And so this really allowed FDA to issue written requests to outline studies that they felt were important to be conducted to improve safe and effective use of products for children. In 2003, PREA authorized FDA to require sponsors of certain drug applications to conduct pediatric assessments. And so there were some additional incentive programs that were implemented, such as the Rare Pediatric Disease Priority Review Voucher Program that was introduced in 2012, and actually currently that is due to sunset in September 2022. There is a bill currently up for review to make that permanent, but that's still yet to be seen whether or not that program will continue. So just to provide a couple of quick highlights that really sort of sets the stage for the RACE Act is really looking a little deeper at what PREA and BPCA has done. So together, BPCA and PREA have actually resulted in over 800 products with specific pediatric labeling information since its introduction. So just quickly, PREA is a mandatory program. It requires pediatric assessment, but the assessment is actually based only on the indication under review. So for your NDA BLA. And is triggered by an application for new indication, new dosage form, new dosing regimen, route of administration, or a new active ingredient. Products with orphan designation are exempt from the requirements of PREA. So it's not just that it's an orphan product, an orphan indication, but there must be actual designation issued. And there are no financial incentives under PREA. With the Best Pharmaceutical for Children's Act, BPCA, it is a voluntary program. Studies are related to the entire moiety, and actually FDA may request studies to be conducted for unapproved indications or different indications from that of the original application. And so FDA can issue a written request, but again, this is a voluntary program. And it really, again, comes back to reflecting the public health need. And also under the written request process, this can be done for products with orphan designation. And there is financial incentives that are associated with the BPCA. Unfortunately, the combination of PREA and BPCA really have not done very much to really impact on development of pediatric cancer therapies. So there was a study that was conducted that looked to really assess what they believe the impact of the RACE Act is going to be on developing products for pediatric cancer. So right now, if we think about drugs between 2007 and 2017, there were 78 adult cancer products that were approved by the FDA, and only 17 of those, which is just about 22%, received any pediatric labeling. And so really, they believe with the implementation of the RACE Act, we can significantly increase the available labeling and for these products in pediatrics. This is just to give you an overview of the products that were available pre-1997, and then those post. And as you can see, there has been, significant advances, but again, it's really the amount of products that have been available for pediatric labeling. So many of the products that were approved in the early 50s and 60s are still being utilized in pediatrics, and many of those actually did not get pediatric labeling until after 1997. So again, right now, with hundreds of cancer therapies available, only less than 40 actually have any sort of pediatric labeling. And there was actually only 4 drugs currently that have been specifically developed for pediatric cancers. So with the RACE Act, it was really a means to encourage overall pediatric development for cancer therapies. So really, the RACE Act creates this platform to accelerate cancer therapies, ensuring that cancer therapies with orphan designation are no longer exempt from PREA. And it really gives FDA the authority to require therapies to be studied based on molecular target rather than indication. Okay. So any original NDA or BLA filed after August, which is coming right up, is subject to PREA if the drug, 1, is intended for the treatment of adult cancer and is directed at a molecular target that is substantially relevant to the growth or progression of a pediatric cancer. And again, as mentioned, PREA applies even if the adult cancer indication does not occur in the pediatric population, because again, it's really focusing on the target modality versus the actual indication, and even if the adult indication was granted orphan designation. Okay, so sponsors will be required to submit their initial pediatric study plan, and that plan must contain an outline of a molecularly targeted pediatric cancer investigation, really targeted at gathering meaningful, quote, meaningful study data. And this is defined as using appropriate formulations regarding dose, safety, and preliminary efficacy to inform potential pediatric labeling. This plan will include any request for a deferral or a waiver with additional supporting documentation. And I'll go into this a little bit more in a moment. So FDA has started to create a framework for defining relevance of molecular targets. And so they have actually outlined some considerations for how they define what are relevant molecular targets. So the presence of a target in one or more pediatric cancers, and really this is not prevalent dependent. So even if it's really only occurs in a small subset, it's really going to be based on available information from the published literature, peer review literature, databases, that there is an appropriate target that could be addressed. And so, again, you can gather non-clinical, if there's non-clinical evidence and other specific information that can help determine whether or not these are relevant targets. So FDA is required to communicate their assessment of regular targets. The statute does direct FDA to establish and publish a list of molecular targets. And what's important to note is that there is actually no specified minimum evidence base. So it's really through evaluation as an ongoing basis. One of the important considerations is that with these lists being published, and this is just an excerpt, and I've provided some links to these for you, is that right now there's over 1,200 relevant molecular targets that are actually listed that the agency considers relevant to pediatrics, and there's 5 non-relevant targets. This is provided as a guide and is really not meant to be binding to the agency. So just because the target is not on the list doesn't mean that studies don't have to be conducted, and vice versa. Just because they are on the list doesn't mean that studies will be required. So each is evaluated on an individual basis. Okay. So I've sort of outlined a process that based on the guidance and kind of the expectations. So as you start to look at your adult program and you're in early phase development and you're collecting data and you're assessing whether or not there's available non-clinical in vivo or in vitro data to really help to determine whether or not the target is relevant to potential pediatric cancer. And then as data is gathered into phase 1, so if there's clinical evidence of biomarkers, again, whether or not there's evidence that you would consider that the target would be relevant to pediatrics. Often it's not going to be clear. And so there's methods for which sponsors can actually obtain consult with the agency to determine whether or not their product is relevant to a pediatric cancer. So there is a method to request a special PDAR IPSP meeting. This is really conducted as a type A meeting. So if you submit your request, you'll be able to meet with the agency within 30 days. You'll need to submit a briefing package and your list of questions at that time. And really they encourage that this is done earlier in the process, because you're going to need to, if you do need to submit an IPSP, that's going to need to be submitted within 60 days after your end of phase 2 meeting. So to accommodate having that interaction, getting that feedback, and then beginning to plan if it is appropriate that you do need to move forward with a pediatric plan, that should really occur fairly early between phase 1 and phase 2. So really, once you have some information, preliminary information of your phase 1, that's really a time where we suggest that you would start that interaction. Sometimes it will be clear that the target is relevant, and you can just then move on to submitting your IPSP. This can actually be submitted before the end of phase 2 meeting to get your initial feedback. But again, this is the relevant process that you'll basically follow. And then once you'll begin to implement your IPSP, then there's an opportunity also for submitting a proposed pediatric study request and to get an issue a written request to move that process forward. So when you begin to think about the content of your IPSP, there are two guidance documents that you should evaluate. One is just the general content of the pediatric study plan, and a newly issued guidance really implementing the study plans specifically for molecular targeted therapies. And really, the plan needs to really outline the objectives of safety, exposure response, and really to define the dose for the recommended a phase 2 dose. And in the exemplar that will be presented by Alyssa, this will become clear as to how this process is actually implemented. Within the content, as I mentioned previously, there will be information on a deferral. Often you'll need additional information in order to then move forward within the pediatric populations from the adult data. So a deferral can be requested, or a waiver, either full or partial, depending upon the target and the indications to be studied. And again, that all falls back to when we discussed the occurrence of various cancers across various populations. So really, just to sum up, the road to success really begins with being informed. So again, recommending that you consult the regulations and guidance documents to assess if the Race for Children's Act applies to your product, and really understanding the needs of pediatric cancer patients, meaning, where are the needs within the population, indication, et cetera, and then really consulting the relevant molecular target list, as well as the non-relevant list, to really begin that assessment process. And again, encouraging that you seek advice not only from subject matter experts, but regulators as early in the development process as possible. And this really will help planning when you think about processes for model-informed drug development to ensure that you're collecting adequate information in adult trials to support its utility in pediatrics. So again, planning, leveraging that adult and safety data, PK, efficacy, to inform your pediatric trial design, assessing the need for age-appropriate pediatric formulations. Again, this is often something that is very time-sensitive, and so that needs to be assessed as early as possible, all driven by whether the age groups that are going to be studied. As I mentioned, planning adequately to avoid any delays in your NDA, BLA in general. To actually reach agreement on a PSP, it takes 210 days, according to the timelines specified in the guidance. So again, that all needs to be planned in advance. And really, it's going to be important to continue to reevaluate your plan as you gain additional knowledge. And this is true for any product, not just pediatric oncology drugs, but again, it's an ongoing process. And as I mentioned, consideration for global drug development. So there are also opportunities, and these are also described within the guidance document, to really coordinate and receive parallel scientific advice. Again, because of the rarity of these diseases, these programs will be likely conducted on a global basis. And it's really important to, again, engage the regulators as early as possible in your development. And with that, I will hand it over to Amy.
Thank you so much, Lynn. We encourage our audience to submit your questions for Lynn, and we'll take a few questions, and then we'll move on to the next presentation. Lynn, it looks like someone would like to know whether we could get a citation for the study that assessed the predicted impact of race on pediatric approvals.
Can you repeat that question? I'm sorry.
This might be one to answer by email. They wanted to know if there was a citation for the study that assessed the predicted impact of race on pediatric approvals.
Oh, I can certainly provide that for you, and we can respond to that on email. I do have that information.
Excellent. And then someone else would like to know, does the IPSP apply to 505(b)(2)s where a change in label is not proposed?
Yes, if it's depending upon why you're, oh, I'm sorry. Does the RACE Act apply? It's the one defining difference with that's important to note between the qualification for race versus general of how PREA is applied is that any new change in formulation, indication, dosing regimen, et cetera, is still applicable to PREA. But under the RACE Act, it's only the original application. So when there's changes to a previously existing application, RACE doesn't apply. It's only the original BLA or NDA where this applies.
Great. Thank you so much, Lynn. And we'll now pass it over to Amy for her presentation.
Thank you, Lynn. Thank you, Suzanne. Can you hear me?
We can hear you.
Yeah, thank you. So today I will share with you various quantitative considerations for pediatric oncology drug development. So children are not small adults. There is evidence from the rapid and continuous physiological changes from neonates, adolescents to adult. An example on this can be seen on the right in the schematic on plot A. Some metabolic enzymes don't exist at birth and mature over time. For example, CYP3A4, it almost reaches adult activities level at the age of 1. However, although the consideration of maturation is important, we should also appreciate not all the data from older age groups will be informative of the younger group, as it will depend on point of disease progressions and mechanism of action of the drug. In addition to the consideration of ontogeny, pediatric oncology drug development also faces various unique challenges. Quantitative evaluation is needed to understand the relationship of various biomarkers with efficacy endpoints. There is sparsity of data due to low prevalence of pediatric disease and limitation of data collection due to blood volume restrictions. We also continuously want to minimize invasive procedures where possible in children, especially when they are very unwell, and ultimately avoid unnecessary clinical trials. And if possible, we also want to understand intrinsic, extrinsic factors such as mutation impact to the outcome of the treatment. Dr. Riemer presented these opportunities and challenges in an ASCPT pre-conference meeting in Pediatric Oncology in 2018. The opportunities certainly can give rise to quantitative approaches that you can see in all the yellow and green bubbles. For example, with the increased understanding of the tumor biology, especially on molecular drivers, and the availability of targeted and immunotherapy as precision medicine. It would enable us to understand the disease progressions and differences of disease in subpopulations such as pediatrics. The availability of big data and emerging of biomarkers also encourages the use of model-informed approach using mathematical models to relate dose, exposures, response relationships, and also using literature informations to inform model-based meta-analysis. And finally, using model-informed approaches to predict very important combination strategies for novel or with standard of care. The quantitative model-informed approach is strongly endorsed by both regulators and industry to support pediatric drug development. You can see shown here are numbers of publications and workshops in the last few years on the topics, and you can go into them to see more details. ISHE 11 addendums also gives considerations when applying model and simulations to pediatric drug development. For example, early pediatric strategy to include multidisciplinary experts of the use of modeling, the data available, and the assumption setting, which is very important. Of all the key questions, those finding in pediatric oncology trials is still present despite the availability of various quantitative approaches. In the two plots in here showing an analysis of 40 molecular targeted agents in pediatric patients from the literature. On the left, the recommended phase 2 dose is often higher than the starting dose that you can see in the pie chart. And this could possibly be due to the uncertainty of the drug exposures and safety of the starting dose. And in such situation, dose escalation will take a lot longer. And on the right, it can be seen that the method most commonly used for the starting dose setting is based on empirical approach of scaling rather than utilizing all the information and with the application of population PK or PBPK approach. There are a range of approaches for scaling adult dose to PK to pediatric population. And in the situation below 1 to 2 years of age, if maturation of the physiology needs to take into account, PBPK, which stands for physiological-based pharmacokinetic modeling, will allow these differences to be incorporated into the predictions. And on the right here, you can see a typical workflow for PBPK outlined, where the starting point is normally a model validated based on adult PK data and the system parametered or physiochem information. Then the physiological parameters are altered to reflect the pediatric age range being considered. And this allows drug exposures to be predicted and so a safe and potentially effective starting dose to be selected. And for older age groups, a weight-based scaling using PopPK may be appropriate. So there are a number of examples in the literature shown in the table here where PBPK approaches have been used to assist pediatric drug development. This ranges from selecting a starting dose in a first-in-pediatric trial to evaluating the factors that might be influencing drug exposures in children to optimizing trial design, for example, on sampling time. Often, we have limited pediatric oncology patients due to the prevalence of disease. We can take advantage of the precision oncology trial designs such as basket and umbrella trials, which was applied quite a lot in adult population, to test new anti-agents, anti-cancer agents in a more effective ways in children. For example, shown here is the iMatrix study. It allows matching of tumor biology with the molecular mode of action of drugs to enroll patients to the trial. And in the particular example of atezolizumab of the iMatrix study, the population modeling using the data collected has demonstrated similar exposure safety profile in pediatric and young adults, and this enabling a weight-based dosing forward. On the right here, other innovative adaptive trial designs with the incorporation of modeled informed approach allowed more efficient dose escalation in phase 1 pediatric trial as well. In the literature with IQ, you can see there is a white paper have published which providing the reveal of the role of extrapolation in pediatric development. There are examples of different therapeutic areas and also the example in oncology. And the paper found that safety data in children is still required in many cases because of the concerns of the potential toxicity of anti-cancer treatments. However, extrapolation of the drug exposures, as mentioned before, can still be very useful to support starting dose selection and dose escalation. And with this paper, it also gives information of how to apply a modeled informed strategy to support a pediatric extrapolation plan. When going into pediatric cancer with a new targeted treatment, the uncertainties of the relevance of the drug's mechanism and the required drug exposures in that cancer. Preclinical models of cancer have supported drug discovery and development for many years in adult setting. With this IMI collaborative effort with ITCCP4 initiative, which aims to develop and characterize animal models for pediatric cancer, and that will ultimately support pediatric cancer drug development through using this data for translational work. It also gives us more insight of how early we can start the pediatric plan in the development and discovery stage. And cancer in children is rare, but present an unmet need due to limited treatment options. The heterogeneity of cancer and the aggressiveness of the disease in children presents greater challenges. Pediatric development cannot be waived based upon the class of treatment if the mechanism is thought to be relevant in pediatric cancer. This means the importance of the drug mechanism in pediatric cancer needs to be evaluated as early as possible, and whether similar drug exposures as adult cancer is required. And therefore, drug development has to be efficient in order to deliver a new treatment to the pediatric settings, or to stop the development as early as possible if the treatment does not show promise. This means efficient trial design coupled with quantitative approaches such as MID3 are required to minimize the number of patients required and time to answer important clinical questions in order to accelerate the treatment available to children patients. These new developments are often brought about through also with public-private collaborations such as IMI and Accelerate. And that comes to the end of my part. And now I will pass it to Larissa and also Suzanne. Thank you.
Thank you so much, Amy. Please enter your questions for Amy into the panel and we'll try and go through a few of them. Someone would like to know whether kinome screens can be used to identify potential undesired targets that would affect pediatrics not identified in adults.
I think we might need to take this question offline, but there is a potential. So yeah, I think I will address this offline if that's possible.
Absolutely. Someone else wants to know what you think the biggest gap is in our understanding of extrapolation to children.
I think toxicity, because as mentioned in the extrapolation white paper, extrapolation to safety currently is not accepted, and also we are still learning more about the relative sensitivity of children to many toxicity versus adult. So in many cases, even we can use a quantitative approach to replace efficacy study. There will also be some post-marketing the requirement to collect the safety data.
Excellent. Thank you so much, Amy. And now we'll welcome Larissa to start her presentation.
Great, thanks very much. Is my audio working okay?
You sound great.
All right, so I will give you a little bit of an overview of the KEYTRUDA Pediatric Development Program and how we were able to support the initial extrapolation label languaging for KEYTRUDA. So by way of general background, tumors use the programmed DEath1 or PD-1 pathway to escape T-cell induced anti-tumor activity, and Pembrolizumab or KEYTRUDA is a highly selective, potent, humanized IgG4 monoclonal antibody against PD-1, which has shown promising anti-tumor activity with manageable toxicity in adults with advanced solid tumors. Pembrolizumab is approved in more than 50 countries for multiple indications at this point. There is a study in pediatric patients. This is the two-part phase 1/2 KEYNOTE-51 study, which was the first study to evaluate pembrolizumab monotherapy for treatment of pediatric patients with advanced melanoma or other PD-L1 positive advanced relapsed or refractory solid tumors or lymphoma. This is just a brief overview of the KEYTRUDA label in the United States. So the initial approval was in September 2014 with an approval for an indication for advanced melanoma in adults. Over the next couple of years, there were additional and expanded adult indications. The first indication with both language around adult and pediatric patients occurred in March 2017. And this was an indication for adult and pediatric patients with refractory classical Hodgkin lymphoma. Over the next several years, additional indications have been added, both for adults and in a few cases with language around both adult and pediatric dosing. So in May 2017, language was added for adult and pediatric patients with microsatellite instability high cancer. In 2018, additional language around refractory or relapsed primary mediastinal large B-cell lymphoma, and there have been some additional indications added since 2018. Those with both adult and pediatric indication language include Merkel cell carcinoma and tumor mutational burden high cancers. So I apologize for the very small text on this slide, but I wanted to show you what the actual label language looks like. What I'm showing you here are excerpts from the KEYTRUDA label as of 2018, because I wanted to show you some of the language that was relevant for the extrapolation approach. If you look at the current label, it's a little bit different because some additional data has become available in pediatric patients. But again, the point of this presentation is to show you how we supported the initial extrapolation language. So you can see here there's some language under pediatric use that efficacy for pediatric patients is extrapolated from results in the respective adult populations. You can see the recommended dosing there for both adults and children for a few of these indications, where it's 200 milligrams every 3 weeks for adults, and then 2 milligrams per kilogram up to 200 milligrams every 3 weeks for pediatrics. And in the pharmacokinetic section, there's some language noting that pembrolizumab concentrations with the weight-based dosing at 2 mg per kg every 3 weeks gives exposures that are comparable to those of adults at the same dose. This is an overview of the pediatric KEYNOTE-51 study. So again, there were two parts to this study. There was an initial phase 1 dose finding part, where a few different dose levels of pembrolizumab were dosed to a small number of children. Then dose-limiting toxicities and pharmacokinetics were assessed. There was a dose confirmation phase where these preliminary cohorts were expanded. And again, there was a dose-limiting toxicity and PK assessment. Also, some IL-2 biomarker data was assessed to get a look at PK/PD in children, and then at the recommended phase 2 dose, there was an expansion into different tumor types to collect some additional safety and efficacy data in children. So the extrapolation language that I've been showing is based primarily on data from phase 1 of the study. The objectives of this KEYNOTE-51 study were to define the rate of dose-limiting toxicities at the maximum tolerated dose or the maximum administered dose of pembrolizumab monotherapy, to characterize the pharmacokinetics of pembrolizumab, to evaluate the preliminary clinical activity within each tumor type, and to establish the pediatric recommended phase 2 dose of pembrolizumab monotherapy. Okay, so in terms of results from the study, no dose-limiting toxicities were reported at the 2 mg per kilogram Q 3-week dose. The observed plasma concentrations of pembrolizumab at this dose were consistent with the PK profiles derived from adult patients administered the same dose. So if you look at that plot down at the bottom of the slide, the dots there are showing the data from KEYNOTE-51 in pediatric patients, and the black line and that kind of beige shaded area are showing you what would be expected, what the expected concentrations would be in adults at the recommended clinical dose of 200 milligrams. So you can see that the data from children with the 2 milligram per kilogram dose overlay quite well with what you would expect in adults at the common clinical dose. The IL-2 stimulation PK/PD results also suggested similar anti-tumor activity of pembrolizumab in adult and pediatric patients, and therefore the 2 milligram per kilogram every 3-week dosing was considered the recommended phase 2 dose in children. So population PK modeling was conducted to support the extrapolation. So what we're trying to do here is basically determine what dose will match the adult exposure across all of the age groups. So the population PK modeling that was conducted was an extension of an adult model that included almost 3,000 subjects across melanoma and non-small cell lung cancer indications. And the model was updated to guide the dosing recommendations for pediatric patients. So it was updated using adults in data with chronic Hodgkin's lymphoma and also in children from KEYNOTE-51. So there were data from 34 pediatric patients with 150 concentration values that were available from that study at the time this was done. In this model, we assessed the impact of cancer indication, age, and body weight on pembrolizumab PK. Parameters from the adult model were re-estimated and refinement was explored, particularly around age and body weight effects. And because there were only 34 patients enrolled in the KEYNOTE-51 study, some additional covariate information from other studies was used to support the simulations. I particularly want to acknowledge here the people who actually did this work, in particular my colleague Ann Chain at Merck, and her colleagues at Certara who helped with this. And you can see there's a page poster reference there from 2018 that has more details of this modeling work. On this slide, I'm showing the simulated exposure by age group for the 2 milligrams per kilogram every 3-week dosing. So there are plots there you can see with the AUC, the Cmin, and the Cmax for different age groupings. So the conclusions for this were that inclusion of PK data from the pediatric and chronic Hodgkin lymphoma studies did not lead to any meaningful change in parameter estimates of the pop PK model. With the pediatric population, there was an age effect with lower age being associated with lower clearance and volume of distribution. But the weight-based dosing regimen of 2 mg per kg every 3 weeks assured similar pembrolizumab exposures across different pediatric age groups down to 6 years. In comparison to adults, exposure was a little bit higher in 2 to 6-year-olds, about 30% higher compared to adults. And exposures in those under 2 were about 2-fold higher. But there is some clinical experience in adults at higher doses of pembrolizumab, so this was not considered a safety concern. Before I end here, I do want to point out that there are regional differences in the KEYTRUDA label. So what I'm showing here is some corresponding language from the KEYTRUDA SMTC in the EU from 2018, where you can see all it says here is that the safety and efficacy of KEYTRUDA in children below the age of 18 has not yet been established and no data are available. So just to wrap up, I hope I've shown you that the KEYTRUDA program used an extrapolation approach where we're trying to match exposures in adults to support initial labeling for pediatric dosing for several indications in the United States, allowing earlier and easier access for pediatric oncology patients who might benefit from treatment with Keytruda. In order to do this, limited PK and safety data were collected in pediatric patients, and population PK modeling and simulation were used to support the extrapolation, and the acceptability of this approach can vary by region. So with that, I'll wrap up. I want to note that parts of this presentation were developed to support a webinar for ASCPT and IQ, and that webinar was developed by the IQ ClinPharm Learning Group Pediatric Work Group, and there's a subgroup within that for oncology and other TA clinical design, which Amy and I are a part of, but we just wanted to acknowledge all the people that were responsible for developing some of the original versions of these presentations. So with that, that will wrap up our content for today, and I think we can open it back up for questions. Thank you.
Thank you so much, Larissa. We encourage everyone on the line to submit their questions to our speakers in the questions panel. Larissa, someone would like to know, how does the current pediatric labeling for Keytruda differ from what you showed in 2018, and why?
Yeah, so some additional data from that Keynote 51 study has become available since 2018. So there was that phase 2 part that was collecting additional safety and efficacy data. So there's a little bit more explicit data about safety and effectiveness having been established in certain cancer types, and some of the extrapolation language has been scaled back a little bit correspondingly. But I just wanted to demonstrate that it was possible to get that initial extrapolation language in there earlier to make the drug available as early as possible to pediatric patients.
Why is the higher exposure in the youngest age group not a safety concern? This is a question for Larissa.
Sure. So in the whole range of clinical studies that have been done in adults with Keytruda, there is some pretty considerable experience at higher doses of Keytruda than the 200 milligram every 3 weeks. And so that's really the supporting data that covers that higher exposure in the youngest children.
Okay. Lynn, someone wanted to know if you could please compare EU versus US regulatory requirements for pediatric clinical studies.
That's actually probably a pretty long response, but in short, the biggest I can say for comparative purposes, it's important to consider timing. So the European requirements for submission of the PIP, Pediatric Investigation Plan, occurs much earlier in development. So that is after completion of phase 1 PK studies, typically healthy volunteer studies in adults. So the plan is developed much earlier in comparison to the US, where the plan is actually submitted after the end of phase 2. Again, when we kind of use end of phase 2 in the US as sort of the marker, but there will be some programs such as what was asked previously on the 505(b)(2), where you may not necessarily have an end of phase 2. So they must be submitted before plan has to be submitted before completion of any phase 3 studies, or must be submitted at least 210 days prior to the planned submission of the NDA BLA, in order to ensure that you have an agreed upon plan that is submitted with your application, because that's a requirement. The other kind of main difference between Europe and the US is really based on the condition versus indication. So in Europe, when you're considering a pediatric plan, that is really based on, it's really based on like a hierarchy, for instance. So it's really the indication. So you can have an indication with a specific indication that is appropriate in adults, but that same indication itself may not be applicable to pediatrics, but there may be another indication that is more appropriate, that is related. So they actually base that on MEDRA hierarchy. So you have to actually consult the MEDRA coding, so you can look at the higher level terms versus the lower level terms, if you will, and that'll help direct the actual studies that need to be conducted for pediatrics. But in the U.S., what really drives what's required for pediatrics under circumstances outside of what we've just discussed for race is that it's really based on that initial indication for your application. So it's whether or not that indication, the indication itself for the NDA or BLA is relevant in the pediatric population. For race, it actually makes it much closer to the European requirements, which is sort of based on this condition. So it's not tied to the actual verbiage of the initial indication, but rather on its mechanism or relevant or the relevant target. I know that's a lot of information, but I can certainly am happy to provide, I have a good resource of a direct line-by-line comparison of the requirements. I'd be happy to provide to the individual who submitted the question.
Thanks so much, Lynn. We've got a question for Larissa. Someone wants to know how receptive the EMA was on the approach that Merck used.
Yeah, so the difference in label language is definitely interesting. I was not directly involved in the negotiations with the EMA, so I'm not sure I can speculate about exactly what happened there, but I know that FDA was very enthusiastic and encouraging of the extrapolation approach, and philosophy in the EMA was clearly different, and they wanted to see some different data to support the pediatric labeling.
Thanks, Larissa. This is a question for Lynn. Someone wants to know whether you suggest that a sponsor submit the PIP and PSP jointly to the FDA and EMA so they're reviewed at the same time.
In my experience, I actually suggest selecting one to negotiate first over the other, because as part of the submission process, you actually will include any previous negotiations from the other regulatory agencies. So FDA requires that you submit any sort of a copy of an agreed upon PIP, and vice versa for the US. So there are avenues, again, because of the timing issues, I usually would recommend the PIP process first, because again, from the regulatory requirements, that process starts much earlier. So that's starting right again after phase 1. Although it is difficult to sometimes comprehend if a pediatric plan that early in development, that is the expectation for EMA, and they will allow you to start or to submit right around the same time as proof of concept studies, but they really want that process started earlier. So typically when I work with clients, we'll work through the PIP process first, and we usually will even start with scientific advice, which I think is really helpful to start there as a starting point. And that process is free to sponsors if they're asking specific questions to pediatrics, and then start to work through that PIP process. And then with the PIP process itself, because once you submit, then you have the 60-day stop clock, so you have an opportunity, and you can get that feedback from Europe, and then you can then start that process depending upon where you're submitting, then start to plan to submit to FDA. So there's a negotiation. So you have a much bigger window also to get to final resolution, where with FDA, you have to, you're really replying within a 90-day, confirmed 90-day window. So that's been my general recommendation for those who are developing global programs where they're going to need to negotiate both the PIP and PSP.
Thanks, Lynn. It looks like we have time for one last question. It's for Larissa. Someone wanted to know if you could clarify whether full or partial extrapolation was used for the Keytruda program.
So in my opinion, the line gets a little bit fuzzy between those terms for full and partial extrapolation. I guess what I would say is that the initial label language was supported on something that seems to me more like a full extrapolation approach, where you have very limited PK and safety data in pediatric patients, and you're really just trying to demonstrate that at the doses that you're labeling for, you're going to match exposure in adults. But subsequent to phase 1 of that Keynote-51 study, that study did go on to collect additional safety and efficacy data in pediatric patients. And so data is continuing to become available, which would, I think, tilt the balance more toward something like a partial extrapolation approach. So it's evolved a bit over time.
Great. Thank you so much, Larissa, and thank you so much to Amy and Lynn and our audience for their attention and all those great questions. Before concluding the webinar, we have a few announcements. On August 19th, we will be presenting the webinar, Diagnostics and Devices, Accelerating Value Communication with Interactive Messaging. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.